really pleased to have with us Dr. Sarah Hernandez, Dr. Rachel Harding, and Dr. Leora Fox, who are the new editors of HDBuzz. And if you don't know what HDBuzz is, that's going to be explained to you. And if you do know what it is, you'll know obviously that it's a research information website, which is very well used in the HD community.
Thanks so much, John. And thanks to the HDA for inviting us. We're really excited to be here with all of you guys. You can probably tell by the sun shining through my window on my face. I'm not in the UK, so I'm somewhere where it's still sunny. Hello, my name is Sarah. And so just by kind of way of introduction, the reason that I got into HD research and my interest in Huntington's disease really stems from a family connection. So when I was about 12 years old, I found out that my grandmother died from Huntington's disease. And so not to totally date myself, but this was a time pre-Google. So it was not super easy for me to get information. And that kind of set me off on a course of just wanting to know more. I had so many questions and I did not have any answers. And really all I could find in biology textbooks was a little sentence, a blurb about Huntington's. It's, you know, an autosomal dominant disease, 50% chance of inheriting it. That was not sufficient for my very curious mind. And that ultimately led to me getting a PhD in biology and postdocing with Leslie Thompson at University of California, Irvine, who's one of the premier scientists in HD research, just because I was curious and I wanted to know more. And then so more recently, I now work for the Hereditary Disease Foundation. I'm the director of research programs there. So I manage the grants programs, I run webinars. So if you haven't attended an HDF webinar, I highly recommend that you do so. I help plan our conferences and I liaise with our scientific advisory board. So kind of all things science and research from the HDF falls under me. And I think I'll pass it over to.
I can go next. Thanks, Sarah. So my name is Rachel Harding. I'm currently based in Toronto, where I have my research team here. But as you can probably tell from my accent that I'm originally from the UK. We were just discussing this earlier. I grew up in Dover in Southeast Kent. And whenever I say Dover, my accent comes back a little bit. It's a very strange phenomenon. But yeah, I studied at Oxford. I did molecular and cellular biochemistry there for my undergraduate. And then I stayed on and did a PhD in structural biology. And structural biology is basically about understanding what really important biological molecules look like in three-dimensional space. And people like me who are structural biologists like to understand how molecules work. And we do that a lot by looking at them and understanding what their shape is and how they might move and function. And so something that interested me about Huntington's disease is that, you know, we wanted to kind of work out how does the Huntington's mutation change the way the Huntington protein itself work? And that's a really interesting research question that people have been trying to answer since the gene for Huntington's was mapped back in 1993. And I've kind of been stuck on this path of really trying to get down to the nitty-gritty of all of that. And I remember meeting at one of my first Huntington's conferences that I went to, and I met Marty McDonald, who's one of the lead scientists in the discovery of the Huntington gene. And she told me that once you start working on this, you won't be able to stop. You'll just need to get to the bottom of it. And she was exactly right. So although I don't have a personal family connection to Huntington's, I've been in this field now for about, oh my gosh, like maybe 8 years or so. And so after finishing my PhD, I moved to Toronto, where I work at an organization called the Structural Genomics Consortium. We do open science drug discovery. And I've been here ever since. I'm now a principal investigator at the Structural Genomics Consortium, also an assistant professor at the University of Toronto. I run my own research team now, and we all work on understanding some of the mechanisms behind like what causes Huntington's disease. And I'm really passionate about the way that we share research, that it should be open and accessible to people, and also accessible in language that makes sense for the people who are most affected. And that's why I'm thrilled to be part of the HDBuzz team. And now I'm going to hand you over to my lovely colleague, Leora.
Hi, everyone. I'm Leora Fox. My sort of journey towards HDBuzz is that I was one of those like high school science nerds who started working in labs really young, but I was also doing some volunteer work in different kinds of long-term care facilities. And so I was aware of Huntington's disease. I grew up in New York City. I live now in Albuquerque, New Mexico, but New York is still sort of the city of my heart. But I studied neuroscience in college because of my experience in high school, and went on to grad school, worked for a little bit, went on to grad school, and basically landed in a Huntington's lab with a really fantastic young mentor, Ai Yamamoto, and I studied mouse models of Huntington's disease. She let me attend conferences and present my work. And what I really loved about the science was communicating it. It wasn't really being in the lab at the bench and having these ideas. And I loved communicating science. So I went on to work a little bit in science communications, and now for the last 7 years, I've been working at the Huntington's Disease Society of America. I oversee our research programs, so funding research, communicating science, and helping drug companies get perspectives from family members as they're designing therapies for HD. And that's me. So I think we can probably start with a bit of background on HDBuzz and how we got to this iteration of the team and who we are and what we do.
Exactly. So,
yeah.
oh, sorry, Leora. So yeah, as Leora was saying, we are kind of the 3 new editors-in-chief of HDBuzz. But many of you might already know that HDBuzz was originally founded and conceived by 2 other scientists called Ed Wild, who is a native Brit as well. He's based in London, and Jeff Carroll as well, who's based on the west coast of the States. And all the way back in 2010, they realized that there was a big gap between the science that was being done and different clinical trials that were being run, and the information that was being communicated to HD family members and the broader patient community. And there were all sorts of very simple questions that people were trying to ask to get good answers, and they just weren't really able to readily find that information. And so they founded HDBuzz with this vision to kind of bridge that and to make sure that people were in the people who needed to know the most, like the biggest stakeholders in all of this research and everything else, which is you guys, were as well informed as possible. And this has evolved over time. The HDBuzz got bigger and bigger, and we're now involved in lots and lots of different activities. But that kind of also put a time constraint as well on Ed and Jeff. And as they've become more senior in their careers, they haven't really had the time to dedicate it as much as they would. So they looked for some bright, young-ish things to come along and take their place. And so that's where they recruited me, Sarah, and Leora to kind of take over the running of HDBuzz and doing lots of writing and editing. So you might have seen since about 2020, when we started doing a lot of this work, a lot of the articles and bits and pieces that we've put out are all either written or edited by us. And earlier, well, like a month ago, Ed and Jeff have officially retired their positions from editors-in-chief, and we've taken over that role, which is very exciting. And we have all sorts of cool plans on the horizon. So watch this space.
And I'll talk a little bit about what HDBuzz does if you're less familiar with it. So the main thing that we do is that we write articles in understandable language about articles about published science that has been peer reviewed. And that's the normal process of other scientists looking at someone else's work and saying when it should go out there and making it public in a scientific way. So we try to take that kind of information and make it accessible to families with HD. We also explain major news announcements. So sometimes there's an ongoing clinical trial and there will be a big data release. And we will talk about that in an article and what it all means and how to interpret it and what's coming next. We sometimes also, if there are stories that are getting overhyped in the news, we can look at those and kind of go back to the scientific literature and say, okay, what is actually significant here, and can we talk about it in a way that's understandable and that really makes it clear what the impact of that science is. Another thing we do is we make presentations. So we, like this one, we do Q&As for different types of audiences, and we also do rather performative presentations at some major family conferences like HDSA's upcoming convention in June. So we'll all get on stage and pick a theme and have all the research relate to that theme. We also all attend scientific conferences in the HD field, and often when we're at those conferences, we are live tweeting the results of those presentations and the science, also in understandable language, and then we create summaries of those on our website. I'll kick it over to Sarah to talk about the support that we have.
Thanks, Laura. Yeah, so another question that we frequently get from people is who funds us and how are we supported? And so that support comes mostly from HD organizations across the globe, such as the HDA, but also organizations like the Huntington's Disease Society of America, Huntington's Society of Canada, and so major HD organizations, Hereditary Disease Foundation. We do not accept money from pharmaceutical companies or sponsors. Researchers that we write articles about their papers, they do not get to see or edit drafts that we write. And we know this is incredibly important, right? We want to remain an unbiased source for Huntington's news for the community. And if we were to take pharmaceutical dollars, or if we were to let people edit our words before they go out there, it would be next to impossible to remain unbiased. And so that's a really critical piece of information for people to know. And another question people frequently have is, what do these funds go to support? Primarily our website. Hopefully you've all been to HDBuzz.net. And if you haven't, don't go now, go after the webinar, but definitely go check it out. And they support travel to conferences so that we can learn the latest and greatest of HD research, so that we can live tweet events. We do a lot of live tweeting, which was mentioned. And also management, administrative fees, writer fees. So kind of a lot of administrative back-end type stuff. And then we recently took over as editors-in-chief, but we have a lot of plans in the works. So we're hoping to kind of build out our website more, see if we can reach out to some areas of the country, areas of the globe that have less representation, build out our website a bit more. And we want to get a lot of ideas from the community. We want to take on new scientific writers, but we also want to have this be a symbiotic relationship with the HD community. So if there's something that you guys think that's lacking from HDBuzz, a space we could fill, a gap that exists, please reach out and let us know. That's something that we really want to make sure that we are there to support you guys. So if there's information that's lacking, if there's a major topic you see that needs to be covered, do not be shy. Feel free to email us at editors@hdbuzz.net. You can find that email address on our website as well. So definitely reach out. And I think that brings us now to the Q&A portion. So ask us lots of questions. We are here to give you information. And I turn it over to John so he can moderate a bit here.
Great. Thanks very much, Sarah. Thank you all for that introduction. Yeah, so we've just as Sarah said, please put any questions that you've got either in the Q&A or into the chat. We've already got a few lined up here for you. So I'll just get started. So the first question it says, "Good afternoon, HDBuzz. Is the expansion of CAG repeats 100% necessary for the onset of disease?"
So maybe I'll kick that off. Short answer, yes. The longer answer is that we've known since the early 90s that Huntington's is a monogenic disease. So that means it's caused by one genetic source. And that source has been named the Huntington gene. And the reason that the Huntington gene causes Huntington's disease is because of that CAG repeat. So without a CAG repeat beyond a specific threshold, the disease will not happen. I don't know if Leo or Rachel has anything to add to that.
I can add that everyone in the world has CAG repeats in the Huntington gene. Most people have about 20, that's around the average, but we know that like over a certain threshold, like Sarah said, it causes HD. We don't know of any other causes of Huntington's disease. It really is necessary to have this extra CAG repeats for the onset of HD. There are diseases that present in the same way, like similar symptoms to Huntington's, but genetically defined Huntington's disease is always caused by these extra CAG repeats.
And do we know what when you get a CAG result, like what it is that that over this number you would definitely develop Huntington's, and below that you wouldn't? What is it that triggers that? Do we know that?
So I think that's speaking about the different thresholds. And so there are these like long jumbled words that scientists and geneticists use about reduced penetrance and full penetrance and all this kind of stuff. But the general rule of thumb is that between 36 and 40, you may or may not develop Huntington's within your lifetime. You're at low risk, but you might carry it on to your children, for example. And sort of 40 and above in general means that you'll develop it within adulthood. Obviously, the longer the CAG gets, after it gets to another threshold where it's even longer, which is again kind of defined differently in different texts. It's normally around 55 or 60. That's where you might get juvenile Huntington's disease. This is where the symptoms begin in childhood or in sort of early adulthood, depending on the length. And so there is this kind of inverse correlation, which means that the longer your CAG number, the earlier you're going to get disease.
And so the next question is, do you have any information on the Sky 0515 small molecule drug trials?
So I can speak a little bit to this. I know that this is, there's a company called Skyhawk Therapeutics that is running a clinical trial of, it's a Huntington lowering drug. I believe it's an oral drug, and it is targeted at the Huntington protein itself. So the idea is that if you have these long CAG repeats, it creates this long protein, and potentially the buildup of that protein and it not doing its job properly can cause harm to brain cells and death of brain cells, which leads to the symptoms of HD. So the idea behind these types of therapies, Huntington lowering therapies, is to try and lower the amount of that toxic Huntington. I don't know whether this is lowering both healthy and harmful Huntington, but I know that it's a type of oral drug called a splice modulator, which makes some little cuts and changes to the RNA in a way that leads to the protein not being produced. I also believe that it is recruiting a very small number of people in Australia only right now. That's all that I know about it. So if you have more to add,
yeah, I think, and also right now, my understanding is that they're not actually recruiting people who have an HD positive gene test. They're just recruiting healthy volunteers in the first run before they then move into HD patient cohorts. So, and that the key thing to know right now is that the trial that's kind of underway or being recruited for is just a safety trial. So this trial, I mean, we hope that everything is safe and it all works out, but it won't actually tell us too much just yet about whether this type of therapy is going to work or what else is going on. But it's a really important first step. You know, we've had like a lot of disappointments and issues in previous trials where we need to know how safe and efficacious drugs are, well, and like any risks that might come along before we do larger trials. So we'll be crossing our fingers that everything looks good in this trial as well.
Thank you very much. Yes, just someone's put in the chat there. Could you ask a question? If you could just type the question, that would be great. So on to the next question that we've got in the Q&A. Someone's asking, is there any effort on behalf of the drug researchers or drug companies in making drugs that are accessible? Not everyone will have access to treatments that are millions of dollars.
This is a super important question. And the easy answer is yes. As we just mentioned, there's there are oral drugs being developed, which are much easier to, usually cheaper and easier to distribute. You know, you can imagine that if a drug requires spinal injections or a brain surgery, then that would be very difficult to do in many parts of the world where people suffer from Huntington's. I also think that we can note that at the CHDI conference, this is a private foundation that supports solely HD research and development of therapeutics. They have a big conference every year, and at the end of February, and this year there were some talks that really emphasized this need to create drugs that are accessible, and really just urge the scientists in the audience to think about it from that perspective. Right now, there's some research going on of really innovative kind of gene therapy treatments that involve surgeries and on other sorts of things. But in terms of keeping in mind that these will need to be accessible, that's something that's being emphasized across the field as well.
Thank you very much. Oh, sorry, Rachel.
All right, no worries. I was just going to say, sort of specific to the UK, is that the NICE is the organization that decides which drugs are available through the NHS. And I think something that, if we get to the point where we have, an amazing drug that is going to slow or halt or even reverse the effects of Huntington's disease, I can imagine that there's going to be tremendous lobbying efforts by clinicians, neurologists, all sorts of folks to ensure that that drug is added to the NICE list so that it would be available through the NHS. I think it's just like we're just not at that stage just yet. But I would imagine that this is something that people are thinking about. And I know I can speak more because I've had more kind of discussions about this with folks in Canada and the US, is that it's a similar, well, the Canadian medical landscape is probably more similar to the UK, but it would be about people are very already gearing up to be ready, that if anything gets approved, that it would be available through, the government healthcare systems, and they're making sure they're lobbying to do that. So you will probably have a call to arms at some stage when, if and when these drugs, well, it's going to be a when, not an if, but some of these drugs get approved, to make sure that the government's aware of the need for the patient communities to have access to these, at minimal cost, which is something which is I think Leora and Sarah have a different situation that they're working with, being based in the States. So yeah, but hopefully on the NHS things will be available when they're working.
Thank you. Next question we've got is actually someone's just wanting to clarify, saying, sorry, did you say that the higher the CAG repeat, the earlier you get it? I suppose that needs a little bit of qualification.
Sure, yeah. So in general, yes, that's true. But there's actually really interesting research that shows that's not the only determining factor for when you get it. And so there's this pretty famous chart in the research world that's like a bar graph that looks like this, if you can see. And then the number of CG repeats are going up and down, and the age that you start to show symptoms is going across the bottom. And there's this kind of curve. So the people that have 180 CG repeats, they can get it when they're 3 to 6 years old. And people that have the more typical 40 to 42 CG repeats, they can really get it between 50 and 60. And so what was interesting, what people noticed is, there's quite a bit of range there between 50, 60, maybe even 70. What's causing that? And so it appears that the CG repeat is the primary driver for when people are getting symptoms, but there are other genes that modify the age at which people start to show symptoms. And so those were cleverly named by scientists modifiers. And a lot of research is going into that right now to define what those modifiers are, to try and figure out if we target those modifiers, can we give people more healthy years? Can we change the age at which they show symptoms? Can we push it off entirely out of the human lifespan? So short answer, yes, CG repeat does correlate to age of onset, but longer answer, there are modifiers that can also contribute to that.
Thank you very much. We've got a question here. So obviously relating to that, are there any studies or research going on that would actually shorten the repeats? And so that follow-up question to that, are the UK and USA working together to share info and meds and studies?
I think for the second question, HD science has no borders. We really, across the board, collaborate with people all over the world. Scientists come together for conferences. There are international collaborations and publications that go out that most universities and researchers have access to. And maybe as a scientist, you want to speak to that a little bit, Rachel.
Yeah, absolutely. I think one of my favorite parts of my job is that every week I'm on phone calls with people in like Germany and Japan, like in all over the US, you know, all across Canada. Canada's a big place, and all across Europe as well. So it's really amazing because you get to talk to people who think about this, and they're world experts in their particular area, like all the time. And we're always thinking about, it's a very collaborative community where people are very keen to work together, share ideas long before they've like submitted their papers for publication. So, which is, you know, in many fields, this just wouldn't happen. So in really competitive fields of drug discovery, people won't share anything. And, you know, it can be quite a closed community. That is not the case at all in Huntington's, I would say. And the other thing that I would say is pretty amazing is that people share things they make in the lab all the time. So like we share things that we make in my lab, so pieces of DNA we've constructed, or samples of the Huntington protein itself, but people give stuff back to us all the time. So Jeff Carroll, who used to be an editor at HDBuzz, he sends me, you know, things that he's made from his mouse models, or different samples of DNA or RNA, and people are very open and sharing, which is really, really amazing. And I'd speak further to the fact that, you know, and I think Leora and Sarah will probably agree with this as well, is that conferences in this field are attended by people across disciplines. So although we're all interested in Huntington's, we have people, nearly every science conference that you go to in the field of Huntington's is attended by people who are geneticists, or they study Huntington's in cell models or mouse models or in people, or, you know, sometimes you'll meet people who are like, they're genetic counselors, or they're neurologists, or they're in the clinic, or they're, you know, all kinds of different stuff. And that's really, really amazing because it brings together people with completely different backgrounds and training and viewpoints to, you know, try and come up with some really good ideas about how we can tackle what's a really quite, you know, difficult, scientifically at least, problem. And that's going to be our, you know, our best shot, basically, having all these super smart people from all over the world invested. And I would also say something that's amazing in most of these conferences as well is the patient and patient organization presence, which I think, you know, sometimes scientists have a habit of getting stuck in their little area of life and not really considering the wider viewpoint. And so having patients present to advocate and remind everyone like why we're doing what we're doing is like incredibly powerful. And it's organizations like the HDA and the HDSA who really develop this and have like enabled these kind of connections to be made. But it's also a testament to the patient population as well, and all the engagement that you guys have, and, you know, being at webinars like this and other bits and bobs is like really, really awesome. But yeah, so very collaborative in all the aspects, I would say.
Thank you very
And
much.
then just
Sorry,
I
Sarah.
can speak to that. Yeah, I'll speak to that first question that was asked. So are there any studies or research going on that would actually shorten the repeats? Yes, is the short answer. I'm doing a lot of short answer, long answer today, but yes. And so there's research going on right now that to look at the genes that are actually causing the expansion of the repeats. And then there's also people, namely Christopher Pearson at in Toronto, who's working on finding small molecules that can induce contraction. So not just stop the seed repeat expansion, but actually induce a contraction. So he's doing really cool stuff. There's lots and lots and lots of labs, academic labs and pharmaceutical companies that are trying to target somatic instability right now. So it's an incredibly hot topic in HD research as of late.
Thanks, Sarah. And actually, that links to the next question, because someone's asking, is somatic expansion the cause of HD? Do we know if drugs like PTC 518 also stop somatic expansion like branaplam?
That's a fantastic question. So is somatic expansion the cause of HD is probably the #1 question in the field of HD research right now.
Sarah,
the answer
would you
is,
mind, sorry, just to interrupt you, would you mind explaining for people who aren't sure what that actually means? Would that be okay, about what somatic?
sure.
Yeah, thank you.
Absolutely. So when you go in to get a test for Huntington's disease, they take a blood sample. They'll tell you, right, like you have 16 and 42 CG repeats. If you do that test when you're 18 or 50, it's very likely to be the same. And that is because your CG repeat in the Huntington gene is very stable in certain cell types. So cell types like blood. The same is not true for other cell types, specifically cell types of the brain. And so there was interesting work by Peggy Shelbourne in the early 2000s that showed in human brain samples, there were CG repeats of up to 1,000 or more in some cell types before those cells died. And those cell types happen to be the exact cell types that are very, very vulnerable in HD. So the medium spiny neurons in the striatum, basically like the very center of your brain. And so these are the cells that control things like thinking and movement and mood, the things that are really affected in Huntington's disease. And so somatic expansion, what happens is there's essentially a stutter. That CAG stutters, the molecular machinery stutters as it tries to read that, and it can get longer and longer in brain cells over time. And researchers think now perhaps this is what's contributing to the vulnerability of those specific cell types, because those are the ones that we see this expansion in. And so right now, what people are trying to do is get a hold on that, understand how we can target that, and hopefully it will have some therapeutic benefit. But that being said, the question stands, like, is somatic expansion the cause of HD? It's a fantastic question. The answer is we don't yet know. A lot of people are looking at this. A lot of people think that it is. Some people think that it's a consequence of HD. We're just not quite sure yet. But what we do know is that the somatic expansion is present in the most vulnerable cell type in HD. So it's not by coincidence. There's something going on with somatic expansion and the expansion of the CG repeat that is linked to Huntington's disease in some way. And I know this is a big topic, so I don't know if Leora or Rachel, you have anything you want to add to that?
I just wanted to add that it's new, right? And so and also, there are human stories in science too. There's rivalry here. There are arguments at scientific conventions. There are papers published by labs with opposing ideas around this and whether it's the cause of HD or not. So we're really still learning. And for, real nerds like us, it's very exciting to see it develop.
But that's exactly how, you answer difficult questions in science, right? Is that, sometimes things are, as humans, we like to partition things into nice little boxes and nice small nuggets of ideas, but often it's actually much more complicated and difficult than that. So having these kind of open debates and discussions and having people present their data and trying to synthesize all of the evidence is really important. And that's really what's, Leora is talking about here. That's how we get to an answer. I would say the other thing that's really critical in this as well is that none of these discoveries on somatic expansion would have been possible without the engagement of patients and family members in research, because a lot of these discoveries have come from either looking at brain samples of folks who have passed and donated their brains and other tissues to science, which allowed scientists to actually discover this phenomenon, but also from DNA samples and other bits and pieces, and being involved with studies like Enroll and all this kind of stuff. So, we're at the point where we're now, we've done all these careful studies with these different and very precious samples. Now we're just trying to figure out what all that data means. And that's what's a bit tricky. But there's a lots of super smart people. This isn't just like, these are some of the best minds in the world. These are folks who are at some of the top institutions, particularly in the US, right? So like Harvard and in Massachusetts and, Rockefeller and all sorts of places who are butting heads and trying to figure it all out in a very collegial-ish way, which is very exciting. So I think we missed part of that question though, Sarah. Maybe we can bounce back to PTC.
Absolutely. I wanted to address that. Yeah, because this is super interesting, right? So PTC 518 is a drug that's being tested in clinical trials right now by PTC Therapeutics. And branaplam is a very similar drug that was tested by Novartis in I believe it was the Vibrant HD trial that ended up being halted for safety reasons. And so both of those drugs are small molecules, so they're taken as a pill. So think like aspirin, and they could pass the blood-brain barrier to lower Huntington. What's interesting is that a paper just came out recently, and HDBuzz just published an article about it earlier this week, is that branaplam not only lowers Huntington, but it does actually affect the somatic expansion of the CG repeats. Totally serendipitous. People did not think that that would happen. That's certainly not why branaplam was designed. It was a Huntington lowering agent. But the reason that it also affects CG expansion is because it's not specific for Huntington. And so this is, as it's called, an off-target effect. Just so happens to very fortuitously affect CG expansion, which is interesting. And you specifically asked about PTC 518 also doing the same. The answer is we don't know. PTC 518 is, I think the compound is still proprietary, so people aren't testing it in the same way that they're testing branaplam in the lab. So we don't know. Probably because I think they're very similar structurally, so they probably do have very similar effects. But very interesting scientific finding that just came out a few weeks ago. So, yeah.
Thank you very much. Rachel, you mentioned Enroll HD. I don't know if you would just mind for people who aren't aware of that, just explaining what that is, because obviously I know it's quite an important sort of research that people can get involved in.
Yeah, absolutely. So Enroll HD is what we call a natural history study. And so what it does is it tracks folks as they get older and as they progress through Huntington's, and sees how they're getting on with different aspects of their life and sort of the physical symptoms and all sorts of other symptoms as well. And I believe that you don't actually need to know your genetic status, right? That's correct. Yeah, sorry, Leora is the expert on all these things. Well, Sarah as well, not to cast aspersions. But yeah, so actually it's something that's great that if you want to be involved in trials for Huntington's to kind of help research along, often for a clinical trial where you're testing a drug, you would need to know your genetic status. But if you don't want to know, but you still want to be involved, it's like a super great option because you don't actually have to reveal that information and it's all kept very private. And it's often like through Enroll and kind of studies thereof that, we have samples and all this data that scientists can mine and dig into and try and find out. See, with the more people you have and the bigger your data set, the more you can kind of extract out of it. And so that's where actually lots of really cool and interesting studies have come from scientists tapping into that data and trying to figure out, what's going on at a kind of more gross level. Is super awesome. And I think there are many sites in the UK. And if you go to the Enroll HD website, you can find one that's close to you and see they have all the information in there about how you can sign up and be involved if that's something that you're interested in or you think that someone else that might be interested in. So we really encourage folks, if that's, if you're thinking about that, it's a really great option for a lot of people.
Thank you very much for that explanation. And the next, I'm gonna put the next two questions together because they're kind of getting at the same thing. So someone's asking, and it's a very difficult question that you may or may not want to answer. Because it's a bit the $1,000,000 question. It's someone's asking, how close do you personally think we are to a successful drug breakthrough? And the related question is, are we close with any of these trials?
I'm happy to start here. In my work at HDSA, I answer or attempt to answer this question all the time. And I don't think my answer is ever very satisfying. But for myself personally, I don't think it's fair to put a timeline. It's not fair for the sake of anyone who's asking, right? All of us here have someone affected by Huntington's disease or we're affected ourselves. Even the three of us on the screen here, I didn't start with a family history of Huntington's disease, but after, more than 10 years in this field, I absolutely have people I love who are affected. And it's maddening how slow research can be sometimes, even as a researcher. So it's very hard to answer the question of how close are we to X, Y, or Z. We know that the trials that are in progress right now are really in the early safety phases, most of them. We're not looking at anything out there right now that's going to be an immediate cure. but there is a lot out there that we are hopeful about. There are trials in progress globally of different drugs that are likely, if they're safe, they're likely to expand. All this new stuff in the somatic instability field, and this idea of shrinking or stopping the expansion of CAG repeats. So the pipeline is very, very rich, but in terms of putting a timeline on that pipeline, research is just it's a choppy ocean. We're all, riding these waves together, right? And it can be immensely frustrating. But I personally would never say like, okay, 5 years from now, this is what's going to happen. Because people have said that to families over and over again, and it doesn't come to fruition in the ways that we always expect.
Yeah, I mean, I would add exactly that, Leora, right? Like, I know when my uncle was first diagnosed, the doctor said, my aunt expressed concerns about her kids, and the doctor said, we have 30 years, we'll definitely have a cure by then. So I think the field has thought this for a long time, right? Like, and Leora's exactly right. To put a timeline on it isn't fair. We can't see the future, but what we can see is the past. And so we've known the genetic cause of Huntington's disease since 1993. And it took us a really long time to get to the first potentially disease-modifying phase 3 clinical trial. But since then, like, honestly, about 5 years ago, maybe 1, 2 pharmaceutical companies at scientific conferences. Now, over 50 at every single conference you go to. There's a lot of hype about Huntington's research, not just within academic researchers, but within pharmaceutical companies. And I think we're getting closer. We can't look in the future, but looking at the past, there seems to be this logarithmic trajectory, right? Like, it's a freight train of research coming to this point in the future. And we can't say when these things are going to be successful, but what we can say, there's this saying that everyone's a shots on goal. We have a lot of shots on goal in the HD space right now, and they're all moving forward with a shocking momentum. And I think that's the best hope that we can have, right? Like, we're looking at Huntington lowering, we're looking at somatic instability, we're looking at drugs that can keep our synapses healthier to maintain connections between the brains. We're looking at a lot of different things. But I think ultimately, when people ask that question, they want something they can do today. And what we do know from research is the only thing that is proven to keep people healthier longer is a good diet and to exercise. And these are super boring things that everyone can do. And they kind of go by the wayside, but there's really a lot to be said for a really healthy lifestyle with exercise and diet. Because right now, that's what we have. But the research is, honestly, it's coming like a freight train. And we can't put a date on it, but I can say that if you look at the past and the trajectory of where we've been to where we're going, it is unbelievable. The momentum is unbelievable.
I think a lot of that as well, the reason there are many monogenic diseases, so these diseases caused by a single type of mutation that's inherited very similarly to Huntington's disease, that are maybe more or less prevalent. But I think the reason that so many companies are really engaged in Huntington's is also because of the community and the organization of the community, right? Which means that if you're trying to recruit a clinical trial for rare diseases and you've got, people, there's not that same level of organization, it's actually really hard even to get like 30 people signed up for a clinical trial with stuff all over. It's very disparate and difficult to pull together. But organizations like HDA, HDSA, HSC, all of these folks who have ensured that there are centers that can host clinical trials, and we've got trained neurologists who can do a lot of the work. And all of that has been funded and supported by HD family members. That is what makes it a very attractive disease for these folks to pursue. And I think, also because these companies also feel the potential benefit that their drug would derive in the same way that the families do. People are very aware that this is a particular area and a particular disease where we really need to get some new drugs approved and into patients. And so people are even across all these different backgrounds, a lot of people in these companies feel quite similarly as well. But it's really, all of this is a testament to the patient community and all the work that's been done by folks present now and advocates past to build this, kind of ecosystem that makes it so attractive to these companies.
Thank you very much, Rachel. And also a couple of people put their hands up and put them down again. If you have a question, if you just type it into the chat or into the Q&A, and we can pick it up. We've got a question here. Are there any potential biomarkers to make trials available for phase 0 to 1 gene positive patients?
I see
Go
Rachel
for it, Laura.
just starting. Well, I think maybe we should first define the idea of this phase 0 to 1. There is a new clinical research tool that was developed in the last couple of years that is a way to stage Huntington's disease. We are all quite familiar, likely, with different stages, for example, of cancer. And that type of staging makes it possible to understand across those stages for all different types of cancer, like what kind of intervention you should do and what kind of treatments work and who should be recruited for different trials. So there's a big effort among many clinicians and scientists in the field to create something like this for Huntington's disease, with the idea of really trying to shift things in earlier disease so that we can intervene earlier and so that we can define people using imaging of their brain and measuring different substances in their blood before symptoms really develop. So someone who's in, it's thought of as 4 phases: 0, 1, 2, and 3. So a person who's in phase 0 to 1 is really in that place where they don't have any symptoms yet. Their phase 0, there's really nothing to distinguish their brain or their body from someone who doesn't have the HD expansion. In phase 1, it's those biomarkers, it's things that we can measure in the blood or through imaging of the brain that tell us that there's something different going on, but there's no symptoms yet, really. So the thing about running a clinical trial is that you have to have something to measure to see whether something works, whether a drug is actually working. So if you gave someone who had not yet developed symptoms a drug for HD and they didn't get sick 2 years later, how do you know if that's because the drug worked or because they weren't going to get sick? And so the idea of measuring these biomarkers is that we can see whether the change that's expected in their body or their brain is being prevented by that drug. So that's the idea of a biomarker. So we have some biomarkers and we have this staging system. One of them you may have heard of is called neurofilament light, NFL. It's something that we can measure in the blood to show nerve damage that might be happening in the brain. But in terms of clinical trials, these aren't quite being incorporated just yet, in part because we need to convince agencies, and I'm going to forget the name of it in the UK, so please help me out, Rachel or John. But we're in the US, it's the FDA. So just to convince the agencies that pass drugs to accept new measurements. So right now, the trials in progress really focus on people who have very early symptoms. The hope is that in future there will be really robust biomarkers, ways to measure and prove, okay, this drug is working before somebody has symptoms. Right now, that is not the case for today's clinical trials, but in future we're hoping that can be measured. If you can measure, for example, like, okay, this drug is stopping the expansion of CAG repeats, then maybe the somatic instability trials within the next few years will be able to incorporate people who are earlier in disease.
Thanks very much, Leora. We've got a question here, and someone's asking, saying, "I'd like to speak to you about mental health as it's important." I don't know if the person who's given that question if you could just say a little bit more about what in particular you would like to ask about that. I'm conscious we're coming towards the end of our time, so if anyone has got any more questions they would like to ask, if you want to pop them in the chat. I've actually got a question from a family member that someone gave me before we started this, asking about research into biotin and thiamine, which was I think something reported on a few years ago.
Yeah, so sorry, Jose Lucas from, I believe, the University of Barcelona. He had some interesting research on biotin and thiamine, and kind of serendipitously, it was because there's another neurodegenerative disease that, ashamedly, I don't remember the name of, and I don't really know much about. But essentially, it's a neurodegenerative disease, very similar phenotypically. So it presents very similar to Huntington's disease, that can be almost ameliorated by the addition of biotin and thiamine. And so it was tested by Jose Lucas's lab in mice, and it was found to improve motor performance in the mice, and I believe thinking tests as well. And so now that's moving forward and being tested in a clinical trial in Spain. And what really lends credence to this biotin and thiamine hypothesis is that it was found completely separately using large data, so omics type, very large samples with a ton of data by a lab at University of California, Irvine. And so anytime researchers can find the same answer that are across the world from each other using completely different techniques, it really lends a lot of credence to that being something of interest. And so that is moving forward in clinical trials. I would caution, because these are things that you can just pick up at like a vitamin shop or something, to not just go out and start loading your body with biotin and thiamine though, because we don't know at what dose, we don't know if they'll actually have an effect. So I think these things are interesting because they are just generally over-the-counter vitamins that people can find. So it's great because they're readily available. But that doesn't mean people should go out and just start loading themselves up with it. We really do want to make sure taking these things is safe and effective before people get them, which is a little bit trickier when they're already available on the market. So that research is moving forward and being tested in clinical trials.
Thank you very much. And we've got some clarification about the mental health question. And I'm just thinking that maybe the person who's asking that, if they want to message me outside of this, because we obviously we can't really unmute to ask more personal questions. So I'll type my my email into the response, so you've got that. We've got another question here asking any updates on the VO659 trial?
This is the trial. Go ahead, Sarah.
Oh, sure. Yeah, I was maybe you can add to it, but I was just going to say this is a clinical trial by Vico Therapeutics. And so what's interesting about their drug is that it targets specifically the CG repeat. And so Huntington's is not the only disease caused by a CG repeat. There's quite a few others. One of those being something called spinocerebellar ataxia. And so they're testing, Vico Therapeutics is testing their drug VO659 on spinocerebellar ataxia type 1 and Huntington's disease in something called a basket trial. So they're kind of pairing those people with those two conditions together and testing their drug at the same time. From what I understand, they're first testing the spinocerebellar ataxia patients, and I'm not sure when they're going to begin testing the HD patients. And I'm not even sure if that's open in the United States right now. I don't know, Lior, do you know if that's being run in the US or the UK, which would be more relevant?
Is that it's just in the Netherlands right now? I would have to look up exactly where it is on clinicaltrials.gov. But yeah, I don't have anything to add beyond that, except that it's they are focusing on they haven't focused on HD patients quite yet. So it's possible that they've got a couple of folks enrolled, but I think yeah, the spinocerebellar ataxia ones have come first.
But I mean, this is a company that's definitely engaged in the HD research community, though. Like, they're present at a lot of these conferences and other bits and pieces. So we're all anticipating that they will, you know, if things look good, and depending on how their trial pans out, like that this will be, you know, something that they might be enrolling people with HD in soon as well. But we just had to watch this space and see how things develop.
Yeah, they'll be speaking at HDSA's convention next week.
Thank you. And I think we've got two more questions that we've probably got just about time for, if that's okay. Someone's saying, hi, I want to ask for some advice from the experts with regards to my dad that's affected with Huntington's. It's quite hard for him due to depression and everything to keep a healthy diet, and especially exercise. The idea of exercising does not fascinate him. He seems bored, prefers to lie down all day. He seems that he can't cut off on some unhealthy food. Is anything I can do or say to help them? I suppose that relates a little bit to what we're saying about. So I think you said, Sarah, about sort of healthy lifestyle, isn't it? It's a bit. I don't know if that's something you were able to comment on or what.
Yeah, sure. I mean, this is tough, right? Like, I would say maybe try to talk to his neurologist and see if there's things that they can recommend. I have to preface this by saying I'm in no way qualified to give actual medical advice. I am not a medical doctor. I do not have a clinical degree. So for any type of medical advice, I strongly recommend talking to someone who's actually qualified to give that. But yeah, I mean, it's tough, right? Like Huntington's affects more than, it affects our mood and depression and causes all sorts of things like that. I would say there's other medications that maybe can help with some of that stuff so that maybe your dad would have more motivation to exercise. But what we do know is that exercise and a healthy diet are two main things that people can do today. They're changes they can make right now that can help them with Huntington's disease.
I think multidisciplinary care is so key. I was just at an educational event this past weekend, and there are some just fantastic social work professionals and, physical therapy, occupational therapy, just wellness professionals talking about, motivation and how the best kind of exercise is the thing that you want to do, even if it's just a walk. If it's something with people, I know that there are a lot of strategies out there around this, but none of us are really mental or physical health professionals. So definitely recommend that you seek more advice on that because there are strategies around this in particular.
Thanks very much. And we've got one more question here. How long does a small molecule like the one Rivka Dickstein PhD found normally take to get to trial?
I'm not familiar with that particular bit of research. I'm not sure if Leora and Sarah are.
Yeah, so Rivka found two small molecules, I believe that lower Huntington, and that was through their interaction with autophagy. So essentially they work by helping break down the expanded protein, if I'm remembering correctly. But I think the question is more along the lines of like a timeline. Honestly, it really depends. It depends on the drug. It depends on how well the research goes. I think it depends on a lot of stuff.
Yep. I mean,
Yeah,
there's a lot of drugs that, or compounds that you can use in a lab that lower Huntington. Like we have many in the lab where you can see if you treat cells, Huntington will go down. But this by no means makes them good drug molecules. This is something where you have a whole different host of things that you need to look at. It's not just their effects, but it's like how they distribute in the body, how they're absorbed, how they're excreted, how long do they hang around in different parts? Do they get to the different organs or tissues that you need them to go to? All of these kind of things. How stable is that drug if you put it in the body? Does it still work? Is it still functional? So you can have a discovery of something that can look like it's will lower Huntington or have another good outcome. But there's many, 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 many steps. At that point, you're really at the beginning of drug discovery. And I'm not familiar with this study as much, like how far along that research went. So I don't know if there's anything you want to add to that, Sarah.
yeah, I know she was in mouse models. So
Yeah,
that's quite a ways away.
Yeah,
there was a lot of like hype about that at the Weizmann Institute, and whether a molecule that works in mice even moves forward depends on a lot of different factors, like where the scientist is and what relationships they have with industry and how well it works when you apply it to human cells and who wants to pay for a trial of that type. And there's so many different factors.
Yep.
So thank you very much for answering that. Just remains for me to say thank you very much to all our speakers. So thank you to Sarah and Rachel and Leora. I think you've answered some really good questions there. We've had some really good some quite meaty questions from the people attending. So we're very pleased that you were able to answer them. And so yes, just to say thanks to everyone who's attended tonight, and thank you again to our speakers.